Okay, yeah, good afternoon, everybody. Welcome. I think it's just gone 4 o'clock, so we'll get started. Welcome. I think I know nearly everyone in the room, but there's a few faces that I'm not familiar with. So if you don't know who I am, I'm Professor Steve Darby. I'm from the School of Geography, but I'm also the Associate Dean for Research in our faculty, the Faculty of Environmental and Life Sciences. And it's my pleasure to be helping to coordinate and organise this faculty inaugural lecture series. I'm not going to say very much, but just to give you the reasons why we run this series, there are really 3 things that I was very keen to try and make sure that we were doing as a faculty. The first and absolutely the most important is that when people are promoted to professor, that is a major accomplishment and milestone in those individuals' careers. And I think it's just really important to recognise and to celebrate that. And so that kind of notion of sharing in that celebration, recognising the research and teaching excellence of our colleagues is just a really important thing to do. That's the first thing. The second thing is you've probably noticed that we usually run a format where we have 2 speakers, a double header, usually with people from different schools. That is deliberate. The idea, at least, you can be the judge of whether this works in practice, is to bring people together who perhaps don't normally or often sit in the same room, and hopefully stimulate some connections and serendipitous conversations. Then the third and final element is that inaugural lectures are funny things. They're quite a particular brief, but they're often reflecting on a set of really cool research contributions that pitch into a sort of a diverse audience. And so there's a sort of an opportunity here to act as a showcase for our research excellence more broadly. And these lectures are, in principle at least, open to anybody, and they are recorded and they are live streamed. And over time, as we've been having more of these, when we put those recordings on our website, we're effectively curating a really nice resource that reflects the diversity of research contributions across the faculty. So I hope that that's evolving into a kind of an element of our public engagement contribution as well. So that's why we're here. I really hope that you enjoy the day. There'll be a chance to enjoy both talks and then the drinks and snacks afterwards. But without further ado, what I'm going to do is invite Diego Gomez Nicola to come down and introduce our first of the 2 speakers today. So thanks, Diego. I'll just hand over to you.
Okay, hi everybody. Steve said we are here to celebrate the career trajectory of 2 of our colleagues, and I have the pleasure of introducing one of ours from biological sciences, Professor Mark Chapman. Mark completed his undergraduate studies in the University of Leicester, and then he moved to St Andrews to change a little bit of the weather, I suppose, experience something different before having a foray in the US, deep parts of the US. And then he returned for final postdoc to the University of Oxford before coming back to us in Southampton in 2013. I think that was fulfilling Mark's dream of coming back to the most tropical parts of the UK, so he could showcase his tropical shirts that he usually presents himself in. After arriving in 2013, he then promoted to associate professor, and in 2024, he finally promoted to full professor. And I have to say, Mark's research is really interesting. I mean, he's interested in how plants have evolved And if we can understand that evolution, to try to identify crops that would give us an upper hand to fight climate change and problems related to food security. So he has instrumentalized that through not only his excellent research, but also excellent outreach and education. So, for example, he has projects with students, school students across the country, which he sends seeds to various people for them to put them into their school pots and see which ones grow, which ones don't. And that would be an essential way of understanding which plants, what crops could be more efficacious, more resilient with climate changing. And this is an essential problem for our society, only highlighted today, for example, in an article in The Lancet about the impact of climate change on not only health outcomes, but also food security. So for those of us familiar with the University Charter, it could be said that Mark embodies the triple helix strategy. For those of you not so familiar with that concept, it could be said that Mark is a box-to-box player. So he's really good in defense, he's really good in attack. I've had the pleasure of working with Mark research-wise, sharing committees with him, and having him around has been always fantastic. He's a fantastic colleague, always ready to speak up and share his expertise. So without further ado, I'm going to leave you with him because I'm sure he has a lot of food for thought for us. Thank you.
All right, so that's my first 10 slides done with. I won't need to go through those now. Sorry, I'll just pause this before I get too shaky. Right, brilliant. Thank you ever so much for everyone for coming. Like Steve was saying, when I was invited to do this, I did think, right, well, I'm going to treat this as kind of like a celebration. So I had 3 beers at lunchtime and I'm good to go. I'm looking forward to afterwards and having a glass of champagne with you all there. So I'm going to give you a kind of a whistle-stop tour of my career so far, not too much about the kind of background, but then what we've been doing in the lab. If ever I say that this is something I've been doing, please remember that this is me in the vicarious sense of actually having people around me doing most of the work. And I just kind of come up with the ideas and then sit in my office and help them kind of troubleshoot. But everything I've done that I'm going to talk about is going to be collaborative work. And I'm going to, at the end, I'm going to give some acknowledgements, but there's people in this room who I treat as very strong colleagues and friends, and I really like working with all of you who I've worked with. So this is my talk. So Steve asked me to come up with the title, and I thought, right, well, I'll put a pun in it. And then I realized about 3 weeks ago that I have to kind of now write a talk about my pun rather than the other way around, which has probably been sensible. But been there, done that, from school classrooms to global food security. And just the start, of course, we can't do anything until we get our funding. So NERC and BBSRC are the 2 biggest funders for my work. Royal Society of Biology have been brilliant with the outreach stuff I've been doing. And the university as well has given me small to medium-sized bits of money to do slightly random things that I've particularly enjoyed, some of which have involved trips overseas, which have been very nice. I'll come back to this picture at the end, but it's a beautiful picture. So this is what I'm going to talk about. So I'm going to go through my journey and what I and we do. I'm going to talk about the beans. These are obviously some examples here. I'm going to give you a bit of a background about food security, so some of the things that sort of set up the projects I've been doing and the reasons why. And then I'm going to talk about 2 kind of facets of work, one of which is called novel crops and one of which is called underutilized crops. And of course, I'll go through these when we get to them. So as Diego said, but I'm just going to go through it again, graduated in year 2000 from the University of Leicester. My undergraduate project was looking at genetic variation in certain species in the UK, trying to work out where they came from, essentially. I did a PhD at the University of St Andrews, graduating in 2004. I then went to Vanderbilt, which is in Nashville, in the southeast of the US. And the whole lab then moved to the University of Georgia after 2 years. So I was the only postdoc at the time. So I was in charge of unplugging -80 freezers, rolling them onto a truck, driving, not me doing the driving, luckily, going for 7 hours to the University of Georgia and then plugging them in and then doing a lot of crossed fingers. But it all worked out and all the samples survived. So I was there until the end of 2010. Start of 2011, I came back to the UK. I worked at the University of Oxford for 2 and a bit years. And then 2013, got my job here. And 2023, got my magic 10 years pin. And 2024, got this hat, which of course I didn't get, but that shows when I became a professor. So there's 2 main things I do in my lab, or we do in my lab. This is the bit I'm not going to talk about too much today, but I'm really interested in plants, ecology, speciation, hybridization. So where do new species come from? How do new species form? Does anyone recognize this by any chance? If you don't, just next time you're at a railway station, just look among all the tracks, and I'm 90% sure you'll see this. It's a species called Senecio squalidus, and it's actually formed as a hybrid. So its parent plants come from Mount Etna. Some plants were moved to the Oxford Botanic Gardens about 200, 300 years ago. They hybridized, they formed this, and it's spread all over the UK. And there's only about 15 to 20 really good examples of hybrid species. So next time you're at a railway station and you spot this, you can be like, I've seen one of the only 20 species that are formed like this. This is another group we work on. This is a more recent project, but again, it's a weedy species in the UK. You've probably seen it if you've never actually recognized you've seen it. And I'd just like to highlight here Richard Abbott. He was my PhD supervisor, really instrumental in the study of hybridization and speciation in plants. Unfortunately, he did pass away about 18 months ago, which was really kind of dawned on me that I really didn't thank him enough for all of his guidance and things like that. So for any of you younger people, if you've got people you look up to, just tell them occasionally thank you, because I wish I'd done that a bit closer. The other part of my research is about domestication and crop evolution. And this is essentially evolution, but going from a wild species to a domesticated species. And the cool thing is that this is a very recent thing. So most domestication happened within the last 10 to 12,000 years. When I was in the US, I studied sunflowers. So this is a domesticated sunflower on the left, a wild sunflower on the right. You can see that there's some quite drastic phenotypic differences. And again, we're talking about very short time frames, a few thousand years. Humans and chimps diverged about 6 million years ago. These probably diverged about 6,000 years ago. So in 1,000th of the time, we've got arguably more morphological differences because of domestication. I'm quite interested in how crops are diversified. So it's not just how you get a crop from a wild species, but how it adapts into different niches. I did a piece of work here, and it turns out if you do a piece of work that you can relate to a very important and well-known piece of art, you can get a slightly higher profile paper. So if anyone else would like to try that approach, please do. But we were looking at the genetics of these types of mutant sunflowers. And again, you may have seen them. You may see them in garden centres. And they're not a million miles away from Van Gogh sunflowers. So that's the way we spun the story. And we got a nice paper out of it. Another thing we've done more recently in Southampton is look at brassica crops. So if you didn't know, most of the brassicas that you eat are actually one species. So all of these, kohlrabi, Brussels sprouts, cabbage, cauliflower, broccoli, and kale, they're all one species. They were domesticated once a few thousand years ago, and then you've got these massive morphological differences into these things that look very, very different from each other. So again, just highlighting, this is a fraction of the time it normally takes speciation to occur. Humans and chimps, 6 million years. All of these guys, less than 6,000 years. So amazing changes you can study. And it's really good as kind of a model for studying how evolution works. The focus of my talk today is going to be the work I've been doing on what I call novel and underutilized crops. And thinking about the crops we can grow in the future and potentially how they can inform us about climate resilience, increased nutrients. We've got a growing population, we need more food, and those sort of things. So I'm going to tell you about food security first. And some of you will probably know a reasonable amount about this, but I'm going to go through it anyway. So 90% of the plants we eat come from just 15 crops. So this is worldwide. 90% of it is primarily from 15 crops. And if we look at all the food we eat, meat, vegetables, grains, everything else, 42% come from just wheat, rice, and maize. So you just look at this pie chart. 19% of the world is eating rice, 18% wheat, 5% maize, corn, and all of its products. So almost half of the calories in the world come from just 3 crops. Now, this is kind of risky. So if you haven't heard of the Irish potato famine, about 200 years ago, there was a pest, a fungus that infected potatoes in Ireland, wiped out almost all of the crop. About 1 million people died and about 1 million people emigrated. So if we're relying on just 3 crops, this is putting ourselves at significant risk. In the UK, we're not quite so reliant on just 1 or 2 things. If you can go to Sainsbury's, you can very easily get anything you want. But 75% of UK agriculture is based on wheat, barley, and oilseed rape. So again, even though we're not necessarily as consumers at such a risk from relying on these things, it is a risk to UK agriculture to be relying on just a few things. And so, of course, the big question is, are these crops at risk from climate change? Probably you can understand where this talk is going, that yes, these crops are at risk from climate change. So this is just an example. So this is looking at wheat, rice, and maize, and soybeans. Soybeans, you might think, well, I don't eat very much edamame, so why does it matter if soybeans are going to be at risk from climate change? Almost all of the meat you eat has been fed soybeans. Almost all of it. Millions of tons of soybeans are produced every year just to feed chickens, cows, pigs, things like that. And this graph just shows the potential drop in yield for a 1 degree C increase in temperature. So for wheat, we've got about 6% drop, rice about 3%, maize 7%, soybean 3 or 4%. So it's a few percent. That sounds quite risky. This is per degree C of increase in temperature. And actually, based on predictions, the climate is not just increasing by like 1 degree over the next 50 years. It's going to be at least 2, probably 3 or even more degrees. So if you imagine that there's a 3 degree C increase in temperature over the next 30, 40 years, that could be a 25% drop in maize yield. I think that's just an example. So these crops are definitely at risk from an increase in temperature. These are the 3 crops we rely on for arable crops in the UK, wheat, barley, and oilseed rape. And you can see that there's a decline in wheat, a definite decline in the last 10 years for oilseed rape, and a bit of an increase for barley. I'm just drawing the lines on that. So we might think, well, if wheat's going down, barley's going up, this is fine. But actually, barley, apart from beer and whiskey and things like that, it doesn't make great bread. And in the UK, we love our bread. And 20 years ago, we were a net exporter of wheat. We produced more wheat than we needed. In the last few years, we've become a net importer. We haven't produced as much wheat as we need to supply all of our industries. So barley is increasing when wheat decreases, but this is largely because if the wheat can't be planted, barley can still be planted later in the year. On oilseed rape in particular has had a particular 50% decline in the last 10 or 12 years. And these are just a couple of quotes. So these are talking about the winter from 2023 to 2024. Difficult weather conditions in the autumn and restricted opportunities for spring drilling have led to a decrease in wheat area. And the rainfall between September 23 and May 24 was the greatest on record in nearly 200 years. So big changes in the climate are affecting what we can grow in the UK. So we are, again, putting ourselves at risk. And at the same time, it's not just about the amount of food. We need to be eating the right amount of healthy food and getting a balanced diet. So there's been various studies that have looked at what we think is a sort of healthy diet, what proportion of this, how many calories come from that, and things like that. And if you plot it as a globe, we're awful. We're really, really bad at eating healthy. Globe-wise, we eat 3 times as much red meat and starchy vegetables as we need. Whereas if you look at fruits and vegetables, it's about half to a third. And if you get down to things like nuts and whole grains, it's 10, 15% maximum that we actually eat that we should be eating. Now, this is, of course, a global picture, and it's not very valuable to say everyone should have exactly the same diet all over the world. But it's important to remember that it's not just about producing more wheat and more maize. that's not going to solve food security. We've got to think about eating healthier foods. And there's an additional risk. So actually, an increase in CO2, which is causing our climate change, is actually has actually got the potential to decrease the nutrients in the things we're eating. This was a seminal paper. It was about 15 years ago now. They did a meta-analysis. They pulled together lots of other people's data. So again, we've got those staple crops I talked about before, and we've got peas and sorghum on there. And I'll just talk you through it. So the two on the left, so this is wheat and rice. They're grasses. They're called C3 because of the way they photosynthesize. And an increase in CO2 will decrease the zinc, the iron, the protein, and the phytate in all of them. Phytate being where the phosphorus is wrapped up in the cells. So pretty much all of these crops are going to be negatively affected by an increase in CO2. Now, these two on the right, the data is a bit more variable because there's less studies, but actually they're probably not going to be as affected as badly. And this is because they photosynthesize in a different way. They're what we call C4 grasses. They have a better way of capturing CO2 and using it more efficiently. But some of our crops, clearly, we're going to have problems. We're going to possibly have less yield, and the yield we get is possibly going to be less nutritious for us. So this is a sort of a double whammy, really, when we think about climate change. Okay. So that's my intro into why all of this is important. So now I'm going to talk about what we've termed novel crops. Now, just to highlight, these aren't novel crops like we generate them in a greenhouse by doing really clever GM stuff or anything like that. We're just talking about crops that could be grown in a region that maybe aren't grown there currently. And one of the things that this is actually, one of the places this has been done in quite a lot of detail is in the UK. So firstly, there was a really influential report that discussed with farmers what they would like to grow, what they do grow, and why there's trouble with some of the things they can't grow. And they suggested these crops would be things that they would be happy to grow. They broke it down into 6 categories. I've just put 3 here. And you'll see that some of them are things that are fairly staple to us. We probably, most of us, I mean, buckwheat maybe isn't top of the list, but buckwheat pancakes are kind of doing a thing now. But sunflower, most of us probably had sunflower oil in something recently, maybe even snacking on sunflowers. Chickpeas, lentils, faba beans. So these aren't things that, you know, we're not trying to change the world by bringing in things that no one's ever heard of necessarily. And farmers are saying, well, actually, I'd give this a try. I would be okay with this. The problem being, when you ask them why they don't, mostly they just say it's not profitable. So I could grow buckwheat, but no one wants to buy my buckwheat. Or I could have, you know, I'd be happy to try lentils, but actually, if I can't sell them, what's the point of me trying to grow lentils? So there's all these reasons, but some of them come down to just not being that kind of market. And so it's really important that we talk to our retailers about the kind of things they sell. And I'll come on to that in a minute. But there's a variety of reasons why farmers just don't grow some of these crops that they say they would be happy to grow. There's crop modeling as well. So if you think about it, you can look at where a crop grows now. It might be Africa, it might be Southeast Asia, it could be North America. And you can say, will that actually grow in the UK in the future? We can model what the climate's going to do. We can say rainfall patterns are going to look approximately like this. The temperature's going to increase by approximately this. What would actually grow well in the UK? And there's a lot of things. So this part of this paper I'm talking about is for an increase by 4 degrees. So it's quite extreme, but it's not insurmountable. We might get there. It's not necessarily a good thing, but you see what I mean. And this is just looking at the potential in the, compared to now, what yield we might get versus what we might get in the future. So these are all crops that are going to have some potential increase in the amount of yield we could be getting from them. And just to highlight a few, so okra, you've probably seen in supermarkets, even if you haven't eaten it. Durum wheat, that's actually a type of wheat that's used for pasta as opposed to bread wheat. We've got sorghum here, was kind of top of the list with it's going to be brilliant in the future. And I just wanted to highlight cherimoya just because it's one of my favourite fruits. If you've never had one, go to the market outside Susu on a Monday. It tastes like custard. I can't stress how good they are. They're amazing. Do have giant seeds in them, so don't just bite down on one, but you know. But all of these crops, we've got soybean in there, sesame, chickpeas, and mandarins. So things we eat now, but that could potentially grow well in the future. So we've got farmers telling us what they'd be happy to grow and what some of the barriers are. We've got modelling that will tell us, well, actually, these crops have got good potential for growing in the future. The problem is no one's really talking to the consumers. And I'm not necessarily talking about most of the people in the room. I'm talking about the consumers of the future. I'm not saying that everyone here is old, because you're not, but some of you are a bit older than others. But what we need to do is we need to talk to the consumers. And so there's this kind of black hole. Because farmers know what they can and can't grow. Then the retailers need to actually buy that from them. Well, the retailers aren't going to buy them if there's no demand from consumers. But no one's talked to the consumers. So we've got a bit of a situation where we're just sitting there saying we know what we could do, but we're not going to do anything about it. Now, luckily, BBSRC put out a grant call that I applied to, and we got funding to build a network to actually partly solve this. So this has only been running since the start of the year. But the highlight of this is that we really want to talk among stakeholders. We've got 2 supermarkets who are interested in our work, who want to come to our meetings. We've got charities who work with young people who want to come and talk to us. We've got a bunch of academics and a bunch of people who can do the sciencey stuff. But at the moment, they're not talking to each other. So building the network isn't about us doing a research project, kind of how you think about a typical grant. It's about getting people in a room and saying, what are the barriers and what are the opportunities? And that's where we're trying to go with this network. And we've called it NUCnet. So NUCs are novel and underutilized crops. And this network is BBSRC funded. We've just got a web page. It may or may not be live today. Different people have found it does work or doesn't work. So thanks to the university for all their help with that. But just to highlight who the team is. So it's myself, it's Jenny and Mary Barker. We're from Southampton. And it's Pete, Lydia and John who are at different places around the UK. And Sandra's very kindly come as well. Sandra is our network manager. This is all our beautiful faces down here in no particular order. So what are our goals? So like I said, this started in January. So we haven't really achieved very much apart from having some goals. But what we want to do is get everyone together who is doing any sort of research or has any interest in these NUCs, these novel and underutilized crops. And we want the research community. And I can say, right, these are the 20 people in the UK who are influential in this field. But they would all be academics. I wouldn't have any idea who else I should talk to. And we want to talk with producers, communities across the UK, generating a network who people can talk to each other, have conversations and discuss barriers and opportunities. These barriers and opportunities are actually going to be ideally turned into some sort of policy. We'd like to talk to farmers more, and we'd like to talk about where this could go in the actual UK food system. We've got money to do pump priming projects, so people can come to us and say, this is the crop that I want to do a piece of work on, and I need a few £1,000s to work with these farmers to do it. So especially if anyone wants to, academically, but wants to work with farmers and school kids, we probably would be very happy to, I can't say definitely, but come to us, we can probably look for funding these kinds of projects. Ideally, these would turn into larger bids and move on to larger projects in the future. And also just developing relationships. So we really want to get work with school kids and community groups to talk about what their interests are, what they would like to grow, what they would like to eat. And that might start to influence kind of the direction of travel of this type of research. Now, before NUCnet, obviously, we couldn't just go straight in and say, we're the people to do this. We had we'd done some research in the past. And one of the things I've been doing is working with school children. And like I said, they're the consumers of the future. So it's really good to talk to, school kids up to the age of any age, really, and ask them what they're interested in and what they find important. And right from day one, I was kind of amazed that they understand climate change. They understand food miles, so where our food comes from. If you're buying green beans and they've come all the way from Kenya, that seems like quite a, you can grow green beans in the UK. Why are we bringing them in from Kenya? They understand these things. They understand biodiversity. They understand conservation. They know why bees are important. These are all really good things to talk to school kids about. So what we did is we said, we're going to send you some beans and we'd like to grow them. And just tell us if they grow. it was quite simple. It started out, it's kind of snowballed since then. But the idea is we're thinking about looking forward 20 or 30 years, what would grow in the UK. And we did this kind of with these groups of crops. So first of all, broad beans, peas and French beans. So these should grow in the UK. So as long as they go in the ground in April or May, they should grow just fine. Chickpeas and lentils. So these are largely grown in the Mediterranean or Canada. They're not widely grown in the UK, but there's a company that has a few farms that does grow UK grown chickpeas and lentils. Whenever they get yield, which is obviously going to be one time per year, they will sell out very quickly, but they are really quite popular with the general public. It's a bit of a middle class thing, unfortunately, at the moment, but hopefully if we can think about the future, we could expand it a bit. They're a bit risky. We can't guarantee to the school kids that they'll grow, but they're worth a shot. And then this is my favourite crop, lab, and we're going to come back to this later. It's very drought tolerant. It's native to Africa. Has anyone here heard of lab? Has anyone, keep your hand up if you've heard of it anywhere else other than via me? Oh, one person. Okay, we're good. Right. So largely, I just go on and on about lab, and I will come back to it, and there'll be reasons why. Hopefully, you'll understand why I like it. But we send them these six types of beans, and the idea being, I want to think about chickpeas, lentils, and lab. Will they grow in the UK? Now, if you send these to the kids and they don't grow, they would be very, very upset, I would imagine. So we had to put in the broad beans, the peas, and the French beans as well. And so we built a website. This was actually just done last year. I've had various undergraduates and PhDs helping me put together these packs. If anyone's free at 11 o'clock tomorrow, we're doing it again this year. But basically, if you have a group of people in a room and you buy them sandwiches and fruit juice, they're really, really happy to do it, which is really nice. This is our map from last year. So this is where all the schools were. The blue is primary schools. The yellow is secondary schools. So we got really good spread across the UK. And then the year before, I did kind of a targeted project with a school on Jersey. And we did, students planted the beans. So we had biology. They analysed the soil. So we had some chemistry. They analysed the pigments using a spectrophotometer that they built, so that was physics. They did cooking, so that was a home ec class. And they did art as well, so all of the beans had these little displays next to them. So we had all of these different classes kind of interested in joining in on one big project. And of course, once you get on the BBC, the university thanks you, so that's really good. This is just some photos that were sent. Now, this is from the first year, so you'll notice that these are all schools that have got these kind of well-established beds, spaces and things like that. And what we realised is that actually there's plenty of schools that can't take part because they don't have this. So in year 2, we earmarked some of the money and we said, if you don't have a garden or space to do this, we'll give you 50 quid. You can go to B&Q, buy some grow bags or a planter and some soil and you can have a go. And there was a handful of schools that took us up on this. And they all said, thank you very much, because there was one in inner city Manchester, for example, who said, we wouldn't have been able to do this. 50 quid is not insurmountable to a school. But for us saying, we'll just do it. Here's the money, just go figure it out. They were really, really grateful. And it was really nice to get that kind of feedback. This photo I really like because these are chickpeas. Really, really healthy looking chickpeas growing. I can't even remember where this is. I apologise. But I was just so surprised how well these crops grow. You can see here there's a sack of peas that came off these plants there. And we asked them for data. So when we do knowledge exchange, you have to remember the exchange part of it. You could. Ideally, you wouldn't just go to a school and tell them things. You need to eat more beans because they're good for you. These are the beans. I want you to grow them. See you later. It's nothing like that. We actually asked them to get involved. And because we have schools from all over the UK, we can actually generate data from all over the UK. So they all filled in a form, they scanned it and sent it back to us. And we got data. So this is the days to germination and the days to flowering. Now, bearing in mind, this is all over the UK. Some of them had beautiful raised beds that they'd been, growing for 5 years with really nice compost. And some of them had like 2 grow bags along the side of a football pitch that were getting hit by frisbees every day. And we still got data. So the staple crops, or the UK crops, the 3 at the bottom, they were the fastest to germinate, the fastest to flower, which is not unsurprising. The next 2 up, chickpeas and lentils. They took a bit longer to germinate. They took a bit longer to flower, but they grew okay. And then lab, again, going back to my personal favourite, slowest to germinate, slowest to flower. And that's not unexpected because it comes from tropical Africa. So this data was not unexpected, but it was sort of unexpected that it worked out quite so well. So I was very happy when we got this. And we also this is data from the second year, but it's basically showing the same thing. We broke it down into, did they sprout? Did they produce a leaf? Did they produce a flower? Did they produce a pod? Again, we've got our sort of UK crops here, so peas, French beans, and broad beans. And the peas and broad beans certainly did pretty well. They all, 70, 80% of them produced pods. Chickpeas and lentils, about 50% produced pods. So they were okay in certain parts of the country. Lab, they all germinated, they all produced the leaf, and about 20% produced the flower, and 0% produced any pods. Which was a shame, because in my garden I got some labs and I had a lovely stir fry with them. But again, this isn't necessarily going to change the world, but it just does show what you can get from a reasonably well choreographed knowledge exchange program. And we got some feedback. So this was quite important. We talked to the teachers and we said, what do you think? How did it go? We asked them some questions and they said yes or no. 16 out of the 18, so quite a small number out of the 100 or so we sent the beans to, but that's okay. 16 out of the 18 said that the project helped them teach new skills or topics. 14 said that the people who took part, largely school kids, but there was one or two kind of scout groups, discussed doing things differently in their everyday life. So maybe their school kids are thinking more about conservation or putting more beans in their lasagnas and things like that. And I thought this was quite interesting, that 14 out of the 18 said that experience with the project increased confidence in using research-based and university-led content in their teaching. And I think this just highlights that maybe there's something that we can tap into here. Because if 14 out of 18 said that it improved this, it's probably meant that prior to this project, they didn't have as much confidence or any confidence in working with a university. So I think this does highlight some of the things that we can do and we can make a difference. So that's the second part of my talk, these novel crops. Third part, underutilized crops. So what do I mean? So there's this really clunky definition, but we put it in a paper just to make sure that we wanted to be clear about what we're discussing. So they're lesser known, they are domesticated or at least kind of domesticated-ish. They're not just wild species. They're locally important. So some parts of the world grow them very broadly. They're part of their local cuisine, part of their diets, part of their customs. But there are barriers to their wider adoption across the world. And we used underutilized to focus on the fact that they have this potential that has not been tapped into. Other research in the past has used words like neglected, orphan, or even just indigenous. And I think this kind of puts a negative spin on them. An orphan crop sounds like it's been a bit abandoned, neglected. It sounds like we've kind of given up on it. Indigenous, that could just mean it's from here. So we wanted to make it clear in our piece of work that we were using underutilized to highlight that there are opportunities that we should be tapping into. Now, this isn't a new thing. So we didn't come along and say, this is our thing, we're going to change the world with it. In the 1970s, these fairly influential books came out looking at specific crops or crop groups and actually talking about their potential for climate resilience and things like that. We'll go back to lab in a second. So there's two main reasons why people talk about underutilized crops and their potential. It's number one, their climate resilience. So it could be drought, heat tolerance, things like that. And number two, their nutrition. So some of the crops we eat aren't particularly nutritious. They just give us the calories. But a lot of these underutilized crops are highly nutritious. So this is an experiment with lab. This is not my work. And really simple experiment. They grew all these crops and then they stopped watering them. And they just recorded when they died. Sounds, if you work on animals, this would be really, really bad. But it turns out if you're working with plants, you can do basically what you want. And lab on the left, it took 30 days before any plants died, and it took 45 days before they all died. Whereas if you compare it to maize and soybean here, and there's a bunch of other things in the middle, it's clearly about, they're lasting about twice, if not 3 times as long without any water compared to these other crops. So already we know that lab is incredibly drought tolerant. Some of the food, some of the food we eat isn't the most nutritious. Now, who here likes edamame, the beans that you get, like the Japanesey ones? Yeah, yeah, quite a few people. Good. So eat those more because they're quite high in protein. But if you can find any of these other crops, these are even better for you. So there's a winged bean, jack bean, moth bean, lab's good, they're still featuring, bambara groundnut, and adzuki beans. So these all have probably twice as much protein in them as most beans that you're eating in your diet. And I asked some kids, have you ever heard of these, thinking that they would all be quiet. And one of them said, I had adzuki beans. And I said, no, you haven't. Turns out they might have done, because you can buy them on Amazon. You can buy adzuki beans here. And actually, wing beans, the Asian supermarket in Shirley had wing beans one time when I went there. So it's not impossible to get these, but they're not exactly parts of our mainstream diet. So lab. So highly drought tolerant is high in protein. So it's got lots of potential at least to be investigated. Even if this crop isn't going to save the world, maybe we can learn about drought tolerance and the production of protein and things by studying it. The first piece of work I did, which was quite soon after starting in Southampton, is there's these two groups. There's two seeds per pod and 4 seeds per pod. So you have these like little squarer pods with 2 big seeds, or you have these longer pods with 4 slightly smaller seeds. And the idea was that these were domesticated separately. Now, if anyone's an evolutionary biologist and you hear about a process happening more than once, it's really interesting because you can study, is it the same genes? Are the same genetic changes involved? Were there any hybridizations, crossing, and things like that? So this is why this sort of piqued my interest. And it turned out, based on doing some fairly simple lab analysis, that yes, it looks like there were these 2 separate domestications. So I was quite excited by that. I had a master's student who's now a PhD student with me, Anastasia, who you probably know, and she studied how drought in lab affects the plant phenotype, how it affects gene expression, and how it affects the nutrients. And I'm just going to tell you a bit about the nutrient work. So first of all, we've got iron on the left and zinc on the right. And within each graph, we've got these 2 are wild and these 2 are domesticated. So the wild lab has more iron and more zinc than the domesticated. So actually, there's breeding resources that we could maybe use to make a higher iron, higher zinc lab. But also, the wild plants are fairly significantly affected by a drought in terms of the amount of nutrients. Not so much the domesticated ones, but that could be because overall they're lower in iron and zinc anyway. So some quite interesting work here, and it just highlights how there are these, going back to the wild and looking at these wild progenitors means that we can find alleles that are potentially beneficial in the future. Here we're talking about nutrients. And I was quite happy to be involved in a project a couple of years ago where we sequenced the lab genome. And in this, we basically had researchers. There was a couple of groups from the UK, a couple of groups from Africa. So we talked to actual crop breeders in Africa, asked them what they were growing and why they were growing it, which varieties are important, which ones are the ones that people like to eat. And we sequenced the first genome. If you look at this circle plot here, we've got 11 chromosomes, which are just shown around the outside. And then each of these graphs underneath just means the different amounts of genes and repetitive DNA within the genome. So this is pretty standard kind of analysis you do when you sequence a genome. But it was quite interesting for lab because lab has quite a small genome. So it's actually quite low in these repetitive DNA and relatively high in its gene content. We did some phylogenetic analyses. So this is looking at how the different samples are related to each other. So to do this, so on the left, we've got a really high quality reference genome, we call it. To do this, we just do we call it resequencing, which is much cheaper and much easier to do. And we group them together based on genetic similarity. So in light green, that's the wild subspecies. And in the sort of darker green, that's the domesticated type. And I'm probably going to advance the slide instead of pointing. Yep, I thought that might happen. Maybe there's... It's the worst pointer ever. So here we've got one group, and here we've got another group. So here we've got 2 seeds, here we've got 4 seeds, and you'll see in both groups there's a wild cluster and a domesticated cluster. So this is really good evidence of 2 origins. We've got a widespread wild species It was domesticated in one place and it was domesticated in another place. So really interesting kind of evolutionary story here going on. And something we could do with the researchers in Africa is they grew hundreds of these plants and they measured all these phenotypes like plant height, days to flowering, size of the pods, size of the seeds. And we mapped onto the chromosome where we think some of the genes are that control these traits. So now we can actually start thinking about potential crop breeding because we could take ones that have the larger seeds. We would know where in the genome the larger seed genes are, and we can do crossing and we can select for the plants that we want based on the part of the genome that's been crossed in. So we're starting to build up these resources that can actually be used to develop lab for the future. Now that was based on 22 genomes. In the past couple of years, we've got generated largely from my lab. We've got about 100 genomes now. So we've got this phylogenetic tree down here, 100 different samples. And these two graphs, they're basically clustering the individuals into genetic groups. So in this one, we've said force it into two groups. So we get a blue and an orange, and I'll explain what's in them in a minute. And in this one, we said cluster it in however many groups you want. And it came up with 7 different groups, although there's only sort of 5 main groups. So just to orient you, so going from the top, we've got domesticated 2-seeded, wild 2-seeded. So these are the ones from the separate domestication. The 2-seeded ones aren't widely grown. So this is basically all of the seed samples we could get. So it was only about 5 domesticated and 10 wilds. The domesticates are only grown in Ethiopia, and Ethiopia has some really strict rules about sending their seed out. So we weren't able to get any more samples of this. So that's kind of just to one side for now. W4, so these are all wild 4-seeded, and then everything else, 99% of it is domesticated types. Now, in the middle, we've got these what we call feral samples. So they're sort of medium-sized seeds, and we're a bit curious about what that would be. But if you look at their genomes, they're sort of half blue and half orange. So they're half domesticated, half wild. If you look at, I'll use the pointer. So this group here, got these intermediate samples, and you'll see here that their genomes are kind of partly wild and partly domesticated. So we think they're recent hybrids. They've maybe cultivated types that escaped a farmer's field and crossed with a wild lab and given this sort of hybridy thing. We've got all of these, we call them early branching because they come at the base of this group. They're all from Africa. So it's really nice evidence that the crop is domesticated in Africa and has then spread. And then we've got multiple times it's spread out of Africa. So we haven't got an African group and a non-African group. We've got non-African samples kind of intermixed with all of our African samples. So multiple expansions out of Africa. And if we just zoom in here, we've got a group, there's lots of non-African samples, all shown in green, but there's Ethiopian samples and Egyptian samples in the middle there as well. So either multiple times it's spread out of Africa, or it's spread out of Africa and then samples have been moved back to Africa. So a bit of a convoluted kind of messy story. But another thing we can do is test for selection. So we can look in the genome and say, do we have evidence of part of the genome being involved in kind of non-neutral evolution, we call it. So anything that's under selection, we're saying is involved in domestication because humans have picked particular plants that have the bigger seeds or the taller plants or whatever phenotype they were looking at. So there's some regions of the genome with really strong signals. And we zoomed in, we've got all these kind of characteristics. So you're probably all thinking, right, let's find out what gene is in there. There's got to be something for big seeds, or there's got to be something for a really important phenotype. Well, there's 30 genes in there. So we're a bit stuck about what we're actually going to do at the moment. And we've got 30 genes in there with a variety of potential different functions. So at the moment, we can't really say which one of those was the really important gene involved in domestication, but something in that region was. I was lucky enough to go to Thailand to work with a collaborator there. This is a lab field growing along the side of the road. The ground was like walking on concrete. It was so dry, but there were plants producing flowers and plants producing pods at the same time. So just to highlight how drought tolerant this crop is. And this is the picture from earlier. This is just the local market. And these are labs. These are white labs, and these are green labs. Someone has painstakingly taken them all out of the pods. And you can see that despite the weather being like this, they were still producing, relatively large crops that they were able to sell in the market. We're doing a project in Thailand at the moment. So these are two collaborators. This is Professor Somta from Kazakhstan University in Bangkok. And this is Professor T.F. Chan, who I've worked with, and he's from Chinese University of Hong Kong. And we're growing out all these plants now. And what we've done is we've crossed different types that vary in their morphologies. And so what you can do is use segregation patterns to actually work out where in the genome the genes are that control certain traits. So this might actually help us track back to our piece of the genome where we've got 30 or so genes, something involved in domestication, but we don't know what. But if we have a seed size allele in there or something like that, we can start making stories about what we think is going on. Another thing we're doing now is we're growing them in the lab back in the UK, and we're looking for these things called nodules. So legume plants, if they're successfully interacting with bacteria in the soil, they produce these nodules. In these nodules, they have bacteria that take in atmospheric nitrogen and turn it into ammonia. Plants can't just take in atmospheric nitrogen and use it, but if the bacteria convert it to ammonia, they can use it. And legumes are the only plant group that can do this. So actually, if you're digging up your broad beans in a few weeks or a month or so, just have a look, and I bet that there's probably nodules on the roots. And so we're doing some microbiology now with Dr. Will King, if you know Will, and we've been growing out the bacteria on these plates, and we're trying to see which ones are actually interacting. And the way we've done this is we've used UK soils, and we've used lab chickpeas and lentils, and we want to see if the UK soils are good enough in terms of their bacteria to produce these healthy nodules. Because without these healthy nodules, you don't get a very healthy plant. It's a bit weak, it's a bit rubbish. I'm just going to give you a whistle-stop tour of a second crop that we've been looking at. So this is something called Kirstein's groundnut. So moving on from lab, but it's another bean. It grows pods underground like a peanut. So it produces tiny little flowers. They grow underground, and then they produce a pod underground. So this is one of the tiny little flowers. These aren't my photos. And then eventually, after about 6 weeks, they'll have produced these pods underground. And there'll be 1 or 2 seeds in each pod. It's used in a variety of reasons. It's eaten, it's drought tolerant, it's nutritious. It's used in religious celebrations as well. And in some places, they actually grind it up and add it to flour to make bread. And that bread has got more protein, obviously, than standard bread. So it's quite interesting the way it's being used. So just last year, we published this. This is the genome of Kirstein's groundnut. Again, this is just another plot, one of these sort of standard things that we all do when we publish. We did the phylogenetics. Now, you remember with lab, there were things kind of all over the place geographically. Well, in Kirstein's groundnut, there's not that at all. Everything groups really well by country. So this tells us that these beans aren't being moved around very much. Everything that's being grown in Ghana stays in Ghana. Everything in Benin stays in Benin. They're not being transported around. People aren't trading them. People aren't growing them in other countries. So there may be some potential there for looking at whether these crops would grow better in other countries, or whether they're super well adapted to where they grow now. But what we did find was a very little genetic diversity. So in doing all of this, we're sort of counting the number of differences we find. We thought, there's not very many. Have we done something wrong? And we ran exactly the same pipeline for Kirstein's groundnut, lab, and cowpea, which is slightly more well-known crop. And Kirstein's groundnut has about somewhere between an 1/8 and the 18th of the amount of genetic diversity in these other 2 crops. So potentially this crop is actually quite endangered, and we probably need to make some concerted efforts in terms of its conservation. What I thought I would do, though, is just see is Kirstein's groundnut odd, or is lab like super odd because it has really high diversity. So we've been doing a small sort of side project comparing genetic diversity in staple legumes, what I've called them, to underutilized legumes. First of all, we sort of looked at what might be correlated, and it wasn't either of genome size or the type or the depth of sequencing. And when I say genome size varies, so normal garden peas, for whatever reason, have a genome that's about 12 times the size of a lab or something like that. Ridiculous variation, and no one really knows why. But we didn't have any, there was no effect of these other factors on the amount of genetic variation. And actually, there isn't a significant difference between underutilized and staple crops, which sort of is and isn't a finding in and of itself. These are just two metrics that we use to measure the amount of genetic diversity. I won't go into it, but you'll see poor little Kirsting's groundnut here has the lowest amount of diversity by both measures by some significant way. So this does really highlight that probably Kirsting's groundnut is a candidate for having to have something sort of important put in place about its conservation going forward. Right, so I'm just going to summarize there. So partly I talked about novel crops in the UK. So we have a network. So if you're at all interested, please join it. We have a website. We've just got a LinkedIn, so please look out for us. If you are related to anyone who is in a school or teaches at a school or is a governor at a school, we would love to hear from you, and we can try and sort something out about growing beans in that school. If you hear about our outreach events, you've got two good things to come for. Number one, stickers. So Anastasia from my lab designed these. No prizes for guessing that came from our Pride event. And we also typically hand out things like recipe cards and things like that, because we do find a lot of people don't know what to do with things like chickpeas. You tell them they should eat more chickpeas, and they say, thank you very much. And then they go away and they say, well, I don't know what to do with the chickpea. But if you give them hummus recipes or pea and mint soup, anything like that, they're actually relatively easy ways to get people to incorporate more legumes into their diet. And in terms of my research, where we're going, so the underutilized crops, lots of people are studying them. So I think we need some way of prioritizing them and thinking about what's the priority things we should study and which crops we should do it in. We should work more with farmers and breeders, thinking about which varieties and which types are the ones that are more important. And something that we wrote about quite recently was that there's this kind of helicopter science, which you may have heard of that term, is people from the West going to these countries, taking the seeds they want, going back to their lab, grow the plant, sequence it, publish their paper. And the poor people who've been growing those for hundreds of years get no acknowledgement or anything like that. So we're trying to do in-country training. So that's part of the thing we've been doing with Thailand, is trying to do kind of online bioinformatics teaching courses, and this decolonialism. So getting people from all over the world invested in these projects and getting some sort of benefit from it as well. So just to highlight, so I've got a few people in bold here whose work I've specifically mentioned today. But in general, I've got my undergraduate, PhD, and postdoc advisors there, bunch of collaborators working on lab, and this is especially Professor T.F. Chan, Hebe, who's in his lab, Praket, who's in Thailand, these other people who've been involved in the lab project, other people all over the world who I've been involved with doing various different projects on various different crops. Anyone who's interested in NUCNet, please get in contact with us, but these are the other 6 people who've been instrumental. And then all the PhDs and postdocs who've been in my lab. Even if they haven't directly contributed to this, they've probably written a script that I've used, or they've probably found some data that I couldn't find, or just fixed something on Iridis that I couldn't fix. So everyone who I have worked with, and a bunch of people in the room as well, I think you know who you are, I am incredibly grateful to all of you. And so this was a team effort, and I'm just kind of showcasing that here. So that's me.
Well, thank you, Mark. That was fabulous. A really interesting, clear presentation on what's obviously a really important topic. We're going to make some time with if people want to ask a few questions at this point, then please go ahead and do so.
Thanks, Mark, for fabulous talk. That's great. I wondered if your favourite plant, lamb, is also resistant to the pathogens. Mainly, I'm thinking nematodes, but any other pathogens?
That's kind of one of the ways. So not specifically me going in this direction, but the people who are doing all of the studies on all of these underutilized crops are doing them in a variety of different ways, using different approaches. So for some, maybe we know about nematodes. For some, maybe we know about the protein content of the seed. For some of them, we might look at drought tolerance. But there's been very little tying up of those things. So I can't tell you off the top of my head about lab. It grew okay in my garden last year. It doesn't necessarily mean that, But that is something we could probably look at with these schools, actually. That might be interesting. Yeah. Okay. Thank you.
I was wondering, in your nut net, do you have any fast food producers, sellers? One of the reasons I want to ask about that is where we are in a university. I remember when my sons went to university, they were quite unusual that they knew how to cook. And basically, all the other students they were living with lived off takeaways. So if you could smuggle these into takeaways, there's a massive market. You get people eating these things without even knowing they're eating them.
One of the so one of the ways you can do it is put beans into bread, and also beans into beer. Which I had some beer that had fava beans in it, which I think was largely just to increase sales, and it was nothing to do with nutrition. But I had it anyway, and it's very tasty. But there are groups like University of Reading, for example, they're looking at reformulating flour for bread by adding 5, 10, 15% fava bean flour and things like that to see kind of how it affects the cooking properties, how it affects the taste, and what difference that would make. And if that could be rolled out to McDonald's, I mean, there's your impact case study kind of thing. The other thing as well is, so I said about those recipe cards. So what we've tried to do is make it kind of 5 ingredients or less, and a bit sort of surreptitiously getting the beans in there. So one of them is kidney bean brownies. So you can make brownies, you can put kidney beans in them. They taste pretty much exactly the same. And there was people taking those cards and saying, I'm not going to show my kids that. And they're just going to go home and make brownies for their kids and see what they say. So there are ways of doing it. I'd rather people were open about it. They don't need to be surreptitious about their beans. But at the same time, I think if you're going to make people eat these sort of things, you need to kind of just get it in there at a sort of low level and sort of build up. But going back to your original question, we haven't specifically, we've got Tesco's interested. So we are going to be talking to Tesco's about kind of how they formulate some of the things. And we could talk about their sandwiches and fava bean flour and things like that. Yeah. Yes. Tim.
If you're looking at the impact of climate change on crop production, food production, is sort of global temperature change actually useful parameters? I sort of imagine, rainfall might be more important, and I guess the local changes in the places the crops grow would be the dominant thing, or is the global temperature kind of correlated with those anyway?
It's probably not. You're right. It probably is very idiosyncratic all over the world. So there are places that are going to have warmer winters and cooler summers, and there are places where it's just going to be chaos. And there's lots of places where we don't know what's going to happen. And if you think over the last 10 years, it's very hard to say what is the climate of the UK, because in one year you could say, well, summer was about 20 degrees and it rained, and then next year it was 40 degrees and it didn't rain for 3 months. So one of the main issues is that there is that unpredictability. And I think some of the crops we need to be thinking of are the ones that can deal with a variety of conditions. But yeah, I think it is very place-based, and we need to think about, right, let's look at the south of England. What could we be growing in the UK in 20, 30 years? Let's look at Scotland. What could we grow? I think we have to break it down into those parts. But it may be we don't know what the climate's going to do. It's just going to be a bit random, unfortunately.
Okay, this will be interesting. We'll let Tom have the last one.
Just picking up on that point, could you do something retrospectively? Because you've sent beans to lots of different schools in the UK. So to sort of look at the weather patterns in the regions, where they can grow, see if you can relate that to their success.
I think that would be great if we had more data. So the data I've shown you has been from a portion of the schools we sent the beans to. And so it would be a little bit scattered, but certainly we could. What I'm trying to do, so what I've done this year is the Royal Society of Biology have given us money, but we've said that we're going to go back to the schools that have done it before, and we're going to try and get kind of like a year on year, and then it might be easier to track. But we haven't tried that, and I think it would be quite interesting even just to have a go, just find the climate data from, weather data.
Okay, I think in the interest of time, we'll leave that there.
Super.
But Mark, thank you so much.
No problem.
Thank you. And in the interest of time, we're going to do quite a quick transition. So I know that some of you will want to leave, but we'll just give it a moment or two, and then I'm going to invite Martin Palmer to come and introduce our second speaker in a few moments.